Dear students, in this lesson we are going to discuss limiting factors in photosynthesis. In the previous lesson we have already discussed the mechanism of photosynthesis. We discussed that photosynthesis takes place in two phases, light reactions, dark reactions, and there we discussed that for light reactions they are the light dependent reactions, the light is necessary for those reactions, and dark reactions we mentioned that they are actually light is not necessary but they are linked to the light reactions, so indirectly they are depending on light reactions. So here the main SLOs are: explain the concept of limiting factors in photosynthesis, predict the effect of varying light intensity, carbon dioxide concentration, temperature on the rate of photosynthesis using graph. So we will be discussing the graphical representation of these limiting factors, the selected limiting factors that are light intensity, carbon dioxide, and temperature. Although water is also a limiting factor, but we are not going to discuss that. Then apart from the theoretical SLOs, in this lesson I am also going to discuss alternative to practical, the ATPs, because they are also part of the final exams. You will be having ATP exam and there you will be having MCQs or objective questions from the selected practicals from the topics you have I already discussed in the class as well. So related to today's topic, these are the practicals which have to be discussed in this lesson. Investigation of release of oxygen during photosynthesis using hydrilla plant, which is an aquatic plant. So this was supposed to be done in the lab as a practical, that during the process of photosynthesis oxygen is released. We were supposed to prove it, but through this lecture we will try to prove that yes, oxygen is released during the process of photosynthesis. And then investigation of light, chlorophyll, carbon dioxide as necessary factors for photosynthesis using destarched plant. We will discuss why they have used destarched plant. So let's first discuss the SLOs, that is limiting factors. What is limiting factor? Any environmental factor, the absence or deficiency of which can decrease the rate of a metabolic reaction, is called limiting factor for that particular reaction. In case of our metabolic process, that is photosynthesis, the limiting factor is simply anything in short supply that prevents photosynthesis occurring at its maximum rate. So what happens when photosynthesis slows down? If photosynthesis occurs more slowly in plants or plant cell, then a lower quantity of sugar will be produced and the quantity of chemical energy available for cell growth will be reduced. Of course, if photosynthesis does not take place, glucose is not going to be formed, and if there is no glucose, then the plants cannot use energy for their cellular processes. So the following factors acts as the limiting factor for the process of photosynthesis. First is light intensity, second is temperature, third is carbon dioxide concentration. We will be discussing light intensity first. Light intensity means the availability of light. For example, you might have observed that two plants, same species, same age, one is placed in dark shadow area and one is placed in bright light. So you can observe a clear difference that the plant which was grown in light, where light was available, it has a good growth rate. The growth is actually dependent on the photosynthesis. So rate of photosynthesis is directly proportional to light intensity. That is, what does direct proportional mean? It increases as light intensity increases and decreases as light intensity decreases. However, at a much higher intensity, the rate of photosynthesis becomes constant. Of course, if light intensity is very high and it is crossing the limit which is actually needed for the cells of plants in order to undergo photosynthesis. So it is not going to increase the rate of photosynthesis if it has crossed the limit. Here you can see on the y-axis there is rate of photosynthesis and on x-axis light intensity. So this graph actually represents what happens. Rate of photosynthesis proportionately increase in light intensity. As light intensity increases, rate of photosynthesis also increases. Here you can see light intensity increases, photosynthesis is also increased, and here at this maximum rate photosynthesis is maximum. But if light intensity is above this limit, then the rate of photosynthesis, this horizontal line represents that rate of photosynthesis becomes constant. Now from this constant rate, in order to increase the rate of photosynthesis, the other limiting factors can affect it. Either the concentration of carbon dioxide or temperature needs to be raised to increase rate of photosynthesis further. So these limiting factors are interdependent on each other. So this is the graphical representation of light intensity on the rate of photosynthesis. Next, temperature. Again, rate of photosynthesis is directly proportional to temperature, that it increases as temperature increases and decreases as temperature decreases. But at a very high temperature, rate of photosynthesis decreases because high temperature results in denaturation of enzymes which are involved in photosynthesis. There are different enzymes which are involved in the process of photosynthesis. For example, in dark reactions, we discussed that Rubisco is an enzyme which is involved in Calvin cycle. So increase in temperature, initially increase in temperature is good for photosynthesis, as photosynthesis increases with increase in temperature. You can see in this graph. Then this is the point where rate of photosynthesis maximum. This is optimum temperature. Usually it is 37 degrees centigrade. So at optimum temperature, but it can vary from species to species. At optimum temperature, the enzymes can perform their activity at maximum. So at optimum temperature, rate of photosynthesis is maximum. But when temperature crosses this limit, if the temperature is greater than this optimum temperature, then the rate of photosynthesis is going to be slow down gradually, because I already mentioned that high temperature is going to denature the enzymes, and if the enzymes are denatured, if denaturation means they have lost their nature, they cannot perform their function properly. If the enzymes cannot perform their function properly, the process is going to be slow down. Moving on, third limiting factor is carbon dioxide concentration. So carbon dioxide concentration rises. As it rises, the rate of photosynthesis goes on increasing until limited by other factors. Again, other factors are going to affect. So increasing the rate of photosynthesis beyond the level, for example, this is the maximum level, like the atmospheric carbon dioxide concentration is 0.04 percentage in the atmosphere. So if it is above that concentration, if it is greater than that concentration, the stomata are going to close and the rate of photosynthesis will be constant, but other limiting factors like temperature and light intensity can increase it further. Moving on, investigation and the practicals. Here ATPs are started. So the first practical was investigation of release of oxygen during photosynthesis using hydrolysis plant. You can read these introduction and apparatus precautions from Punjab textbook as well, because these experiments have been given in your textbooks as well. But here I will just briefly explain it, and then I will attach videos and you can get those videos. I have already uploaded the videos in GCR classroom, and you can take the videos in your USB as well. So I will just explain it briefly. So the introduction is that of course, the end product of photosynthesis is oxygen. Oxygen is a byproduct in the process of photosynthesis. Through this experiment, we need to prove that yes, oxygen actually it is released during photosynthesis. In order to do that, we need to have a special setup. In this diagram, you can see there is a beaker. You can fill it with water, about 500 ml water, and you have to dissolve sodium bicarbonate as a source of carbon dioxide. You can also add potassium bicarbonate because both the molecules release carbon dioxide, because in water, if we perform this experiment, it can be deficient of carbon dioxide, which is one of the limiting factors. So we add sodium bicarbonate as a source of carbon dioxide, and then we put the hydrilla plant, which is aquatic plant, under a funnel. We take a funnel and we invert a funnel over this plant, and then above the funnel we have placed a inverted test tube, and we place this setup under sunlight. We expose it to the sunlight and wait for some time, like 2 to 3 hours. Then after some time, observe what happens. Of course, if carbon dioxide is available, if water is already available in the cells of this plant, if light is available, then the process of photosynthesis is going to take place. And when photosynthesis occurs, it will produce oxygen, and the oxygen is produced at the end of test tube. It will produce, and you can also observe the bubbles. Through the bubbles, gas bubbles are seen here, and the oxygen will actually accumulate on the top or the bottom of the test tube. And in order to test it, we can just take out this test tube and insert a glowing stick or a matched stick into the test tube. If it remains burning, then oxygen is available, because you know that oxygen is important gas for the process of combustion. So through the bubbles we can also observe, and through testing with a burning stick or glowing stick, you can also check the availability of oxygen here. Next experiment is investigation of light, chlorophyll, carbon dioxide. So 3 things are there. We have to discuss one by one as necessary factors for the process of photosynthesis using D-starch plant. So first, let me tell you what does D-starch mean. D-starch means plants, when they have undergone the process of photosynthesis, they make glucose, and glucose is stored in the plants in the form of starch. Starch is a large carbohydrate. So if the plants have already, the photosynthesis has taken place, they have stored starch in them. So in order to perform this experiment, first we have to remove the starch. So removing the starch is actually D-starch. So how we remove? Let's do for light as a necessary factor. Select a healthy potted plants. We are using potted plants because we can easily, they are portable. We can take them into the room, out of the room, whenever it is according to our requirement. So select a healthy potted plant. De-start the plant by placing it in complete darkness for 48 hours. Like if you put a plant in complete darkness for 48 hours, it is going to use all the available starch during the cellular process, that is respiration, in order to get energy. So the plant has used all the starch within these 48 hours, because it has not new photosynthesis or the photosynthesis has not take place for 48 hours, and the available starch was used by these plants in the leaves. We are talking about the leaves. So they have used the available starch. After 48 hours, using a clip or cellophane tape, carve a part of the leaf of the plant from both upper and lower side by black pepper strips. We will cover it in this diagram. You can see we can select any one leaf from that potted plant, and we will cover some part of that plant using clips. We will attach a black pepper. Black pepper has the ability to block the sunlight. It is a special pepper which blocks the sunlight. So the whole leaf is actually de-starch. Now we are going to expose it into sunlight in order to check that light is important for photosynthesis. All the other limiting factors are available, like temperature is available, carbon dioxide is available, but light is available to some part of the leaf, and here in this part where we are covering with black pepper, light is not available. So what we expect, in these cells photosynthesis will take place, and where light is not available, photosynthesis will not take place, and we will prove it through the experiments. See what we are going to do. After 3 to 4 hours, pluck the leaf and remove the black pepper. Now we will check the presence of starch using iodine test. In order to do that test, we have to remove the chlorophyll, because iodine does not give color with chlorophyll. So in order to do that, boil the leaf in water for 2 minutes and then decolorize. To decolorize the leaf, transfer it to the boiling water to a beaker containing ethanol, and ethanol actually dissolves all the chlorophyll from the leaf. Starch is still there. Now remove the leaf from the beaker and wash it with water at room temperature. Place this leaf in a petri dish. We will take a petri dish and we will place the decolorized leaf in the petri dish, and then we will perform iodine test. We will put few drops of iodine. The area of leaf that was uncovered performed photosynthesis, and we will have starch, of course. Therefore turn blue. You better know that iodine is actually a brown colored solution. It gives blue color with starch. So these portions, they will give blue color, and the covered portion, this one, the middle part, will remain yellow, as it does not perform photosynthesis, and it is not going to give any color with starch, because any color with iodine, because starch is absent there. I will attach the videos, and it can be more interesting for you people. You will understand it more clearly through the videos as well. Moving on, next is chlorophyll. You can read it through the PowerPoint presentations, which I have already uploaded in the GCR. Here I will explain with the help of diagram. First of all, chlorophyll is the green pigment, right? So we will take a variegated plant. This is a variegated leaf, right? Variegated means it has, in nature these leaves are found in different trees, like half portion is green and half portion is automatically yellow. The yellow portion means it is deficient of chlorophyll. Chlorophyll is not present there. So we select a variegated leaf in order to check that chlorophyll is necessary. Place a variegated leaf in boiling water to kill and preserve, because we place a variegated leaf in boiling water and then we will transfer it 10 minutes in hot ethanol boiling test tube so that chlorophyll is removed. The leaf is dipped in boiling water to soften it, because ethanol makes it harder. The leaf is separated out in petri dish and covered with iodine solution. The area that had chlorophyll gives blue color with iodine, and the area that had no chlorophyll remain pale. See, first the variegated leaf is boiled in water, then it is shifted to a test tube containing ethanol. You can directly heat it, but ethanol is combustible, it can burn. That's why we put a beaker with hot water and we put the test tube containing ethanol, and inside that there is the leaf. This is called water bath. We give it a water bath, and after that we dip the leaf, we remove it from the test tube and we dip in water in order to soften it. Then we place it in a petri dish and we perform iodine test, and iodine will give this area which was green, this area will give blue color with iodine. Area affected by starch is blue test, because they have starch is present, because chlorophyll was present, means photosynthesis has taken place, and proof of photosynthesis is that they have starch. So it will give blue color with iodine, and this part will not give blue color. Moving on, carbon dioxide. To prove the necessity of carbon dioxide in photosynthesis, following steps are required. Insert a part of the leaf of a discharged plant. Again, discharge plan, how you can discharge, by placing it for 2 to 3 days in complete darkness. We will This is a discharge plan, and we will place half portion of this discharge plan into a conical flask containing potassium hydroxide. Why we have used potassium hydroxide? Because we need to check the necessity of carbon dioxide for photosynthesis. In this jar, potassium hydroxide is acting as a carbon dioxide uh, absorber. It absorbs the available carbon dioxide in the jar. So in this jar there is complete absence of carbon dioxide. See, potassium hydroxide solution absorbs the carbon dioxide gas from the air present in the glass bottle. Leave the plant in sunlight. After few hours, perform starch test. After few hours, this was the part which was inside the jar, and this was the part which was outside the jar, and this is complete airtight, means this portion gets carbon dioxide and this portion does not get carbon dioxide. So when we perform starch test, this portion which was outside the jar, it will give blue color because photosynthesis has taken place, and in this part photosynthesis has not taken place because it was inside a bottle, inside a jar where carbon dioxide was absent, and absence of carbon dioxide means no photosynthesis. No photosynthesis means no starch, and no starch is proof using iodine test that iodine does not give any color. It means photosynthesis has not taken place. So this was all about this lesson. Hope you will understand these practicals more clearly through the videos which I will attach. Thank you for listening.